Um, okay, we're live. Hello, everybody, and welcome to another session. I hope so far you have enjoyed your talks, and I have the pleasure now to welcome Dr. Krishnamurthy from the Scripps Research Institute in California. So we'll quickly introduce Ram. Dr. Krishnamurthy studied chemistry at the Indian Institute of Technology in Mumbai. Then for his PhD, he went to the Ohio State University in Columbus in the group of David Hart, which had a focus on synthetic and mechanistic organic chemistry. And it's then during his postdoctoral fellow at the ETH in Zurich that he discovered his passion for molecular evolution, attending a talk by Professor Eschenmoser about why life chose ribose over hexose for the genetic material. Later then, he worked as a NASA NSCORT fellow with Gustav Arrhenius at the Scripps Institution of Oceanography. So Arrhenius himself, who is a family with 3 Nobel laureates, was a geochemist who studied crystal chemistry, cosmochemistry, and prebiotic chemistry, and worked to plan the analysis of lunar samples. So this helped then Dr. Krishnamurthy to gain further insights into evolutionary chemistry. So he then stayed in La Jolla as a senior research associate and later as an assistant professor at the chemistry department at Scripps. And since 2005, he's an associate professor at the institute. His talk today, Chemical Origins of Life and the 18th CAMEL Paradigm, will consider some interesting questions regarding molecular evolution. Thank you for joining us today, and the online stage is all yours.
Okay, thank you very much for the kind invitation and the nice introduction. So let me jump straight into the presentation, and I'm going to start sharing my screen. And let me know if things are fine or not. And off we are running. So there we are. So can everybody see my screen? Is everything okay? Can people hear me?
Yes, we can see it.
I'm not hearing any feedback. That's why I'm just waiting.
We can see your screen.
Okay, good. That's all. I just wanted a confirmation. So I'm going to begin presentation. So what I want to tell you today is this story of chemical origins of life and the 18th CAMEL paradigm. And during my presentation, I will introduce you to a chemical puzzle, a mathematical puzzle. And please don't Google it during that time. I'll tell you the answer, and I think you will enjoy the answer that comes up with. And I'll tell you what the connection is. So what we are interested in is, how does chemistry become biology? How does a mixture of chemicals come together and give rise to molecules, which then begin to interact with each other and give rise to a property we call life? And for us, this is a problem of transition. For example, how do most of us, or at least largely most of us, believe in this chemical, Darwinian evolution, where a small organism becomes this highly evolved human being. Now, the reason we can do that is because we can find fossils with which we can go back and study these, how evolution would have occurred. Now, this is simple, but when you replace these, things with, I would say, chemicals, what happens is that, there is prebiotic chemistry. And then the question is, how does prebiotic chemistry give rise to what we know as biology and life? And it's based on these molecules. And prebiotic chemistry is very good at coming up with these kind of molecules. You find them in meteorites, you find them in prebiotic simulation laboratory experiments. But then the question is, how does that become these kind of polymers and self-assembled supramolecular structures, which then go on to give life? And so we have now this gaps of knowledge. How it happened, we do not know. We are trying to find out. Now, there are also black boxes, and black boxes in the sense that even if I do demonstrate in my lab, a pathway from amino acids all the way to a protein that is functional in life, you will always ask me the question, do you know this is exactly how it happened on early Earth? And I have no way of answering it, because the early Earth has kind of destroyed its history. And, you can't go back and find chemical fossils by which this pathway exactly happened. And therefore, we are not able to construct it in a way in a historical fashion. But our duty is not to really go back into history, but to show that it can happen. Not whether it did or could, but it can happen. Okay, that's our way of doing it. Now, this is where I want to introduce this 18 camel problem. So this is a story that comes from the Eastern culture. So there's a father with 17 camels, and he has 3 sons, and then he passes away, and his will says the half of 17 should go to the first son, 1/3 to the second, and 1/9 to the third. And you cannot do this math and have a happy camel or a happy family. So the question is, how do you go about doing it? And I'll tell you the answer a little later, but let's go back to the problem in hand. In our area of work, we also have these kind of camels. They are called, prebiotic chemistry reactions. For example, this is a very famous Butler-Formose reaction, where formaldehyde in the presence of a strong base gives rise to many sugars. One among them is ribose. There is the famous Urey-Miller experiment, which gives rise to amino acids starting with gases by a spark discharge experiment simulating lightning. And then that's the Strecker reaction, very famous Strecker reaction. And then there is the John Oro's observation that if you take hydrogen cyanide, it can polymerize to give black polymers, which when hydrolyzed gives rise to many heterocycles. One among them is adenine. So this sort of simple chemistry catches people's attention and says, look, if such simple molecules which could have existed on earlier give rise to these molecules which are in life today, life must be very easy to happen. Okay, and this is now reinforced by the headlines that you see when molecules are found in meteorites. For example, this is the famous Urey-Miller experiment, where they found that the experiment matches very closely with the distribution of amino acids in meteorites. Then you find sugar-like compounds in meteorites, and then you find these nucleobases in meteorites, and then the headlines scream life molecules found in building blocks found elsewhere. But as a chemist, if you're realistically looking at it, the conclusion that you come to is that these molecules, the chemistry leading to these molecules are universal, but not life. Life is not just a mixture of chemicals that you put together and voila, you have it. That doesn't happen. So, but this kind of observations has given rise to, historically in this field, different first approaches. That means one of them was a protein world. That is one of the most ancient, when people found that proteins could, amino acids could be formed so easily, and they are so universal in meteorites. They said it must be that they gave rise to proteins, and proteins somehow gave rise to RNA. Then you went on. So that was the protein first theory, which proteins came first and then the rest followed. And then, of course, there is this RNA world, which says, no, RNA came first, a pure RNA molecule which could self-replicate, and then the whole thing took over. Protein was invented later, then protein and RNA invented DNA, etc., etc., etc. But then there are people who believe that metabolism came first, because there are small molecules that interact. They somehow form cycles of reactions which then evolved to give rise to building blocks of RNA, DNA, and proteins. Then there's, of course, the lipid world. People say, look, you need to have a lipid in which everything is enclosed. Without that, the reactions would not happen in an ocean where you would get diluted. Therefore, you need this. So there's all this me first theory, and then everything came later. But slowly, this kind of approach is fading away in this field and giving rise to what is known as systems chemistry. And I will talk about it a little briefly, later in the section. So this is what you have. So there we have our 17 camels. And there we have the will. Don't ask me whose will it is, but it's the will of the father in that story. And there is biology and life. So how do we get from here to here? So people like us who work in prebiotic chemistry, we try to build the track from bottom up. Then there are people who are in biology who build it down. That means, how simple can a cell be? Do you need all this translation machinery? So what you do is you try to remove certain components and still see if the cell functions. So the question is, can we have a happy meeting, somewhere in the middle, where the prebiotic chemistry will transition into that simplified, cell-like structure that can go on to biology? But so far, we are struggling to make the ends meet, if you will. And if I publish a paper and people come and say, no, Ram, you used a condition that is probably not on earlier. So what I do is I try to adjust it. So I try to adjust that one condition, but then I find that all other of my results go out of connection. So that means there is this constant struggle in this field, how to make this transition from chemistry into protobiology or biology. And I was trying to search for a figure, to say that probably they don't meet very well. But when I came across this figure, it gave me an insight. And actually, probably we are wrong in trying to run from one station to the other station in the same train, but maybe we have to change stations in the middle. There is no direct connection. And this is not a very far out idea, because there have been changes on early Earth with very violent changes in atmospheric composition. There is the great oxidation event about 2.5 billion years ago, where there was no oxygen on early Earth. Suddenly, a lot of oxygen appeared, and the chemistries that could not adjust to that oxygen had to die off, and then you had to adjust yourself to the oxygen, and so new chemistries had to be invented. So some chemistries died off, some chemistries continued, but new chemistries had to be invented, like oxygenic photosynthesis. So if you follow only oxygenic photosynthesis, you will end up here and you will not proceed further. So then what do? So you continue, and with that, you ask the question, how do I get RNA from this mixture? Only by focusing only on RNA specific building blocks, or are there other candidates that you do? And both approaches have their strengths and weaknesses. I will not go into them, but what I want to point out is, if you focus only on biology to guide you, and I'm going to show you how focus on biology only trying to get information to prebiotic chemistry can mislead you. So for example, let us take RNA. And you're all familiar with that. And there is the pyrimidines, where you have the cytidine and the uridine. And then that is connected to the prebiotic chemistry that I showed you before, where you can have, cytosine and uracil. And then people make the connection. Look, there are the molecules, therefore somehow they must be connected, right? But if you look at biology makes these compounds through orotidine, and orotidine is made from orotic acid, and orotic acid comes from aspartic acid. Till today, biology does not make any of the nucleobases de novo. Okay, what it does is, the only base it makes de novo is orotic acid. All the others are either through a salvage pathway, as you know. The only nucleobase it makes is this. And incidentally, prebiotic chemistry also gives you this. But people are not worried about orotic acid. They only focus on uracil and cytosine, or the adenine guanine, because this orotic acid is not finally present in RNA. So what you're doing is, you're looking at the final product, and then you're looking at the starting raw materials and saying, how can I connect them? Forgetting there might have been some intermediates that do not exist in the final product. But case in point, in biology, you have orotic acid. That's the only nucleobase that is made, but then it's not in the final product in the same form. So we've started wondering, why is orotic acid not in RNA? Why do you have to lose this carboxyl group to go to uridine? So we made RNA containing oligonucleotides. This is a thermal melt study. Many people might be familiar, to study the stability of the duplexes hybridization. And what we found was, here is the regular, the black one is the regular thermal melting curve of the RNA. And if you replace one of the U with orotidine, you see you lose that stability. That means you do not form the Watson-Crick base pair duplex anymore. So that means orotic acid is not functional, and it's as bad as a purine-purine mismatch, not even a pyrimidine-pyrimidine mismatch. And the reason we understand is why, because what orotidine does is, for a Watson-Crick, you need your hydrogen bases, bondings on, the Watson-Crick phase. But because of the steric hindrance, it turns around, and what happens is you have now the Watson-Crick facing the wrong way, and therefore there is no base pairing. So you might ask the question, so what's wrong? Nature chose a better base that is Watson-Crick functioning. Doesn't nature do that? And the question is yes, but you had to wait to make the choice till you made the oligomer. You couldn't make the choice when it was just orotic acid. So you had to make it only when you found there was no base pairing. So the question then becomes, when you are making choices at the level of an oligomer, how can you already decide just based on the meteoritic distribution of adenine life? You can't. So the question becomes, How do I know to select the molecules here in a mixture when I don't know what the end product is? Okay, how do I select? Do I select it here? Do I select it here? Do I select it here? Or do I select it at the level of a function that is useful to the organism? So, there are different levels of selection depending on what the properties are in a given environment. And I can't make a selection for a molecule that has not yet appeared. Because if I try to do that, then it's teleology. Because I already know the outcome, I can reach back. And remember, in our type of science, there is no teleology. Because, as somebody famously said, evolution is a tinkerer, not an engineer. And that is also true for chemical evolution. So, the model that we are following in our lab is that we don't, look at what biology is doing. We just start from mixtures and let the mixtures teach us whether we can get to something functional, just by the properties that emerge as the mixtures are being formed. So the question is, can you, imagine RNA coming out of a complete mixture of materials? That's the question, and that would be a non-teleological approach with biology. So we do not know whether that RNA is the outcome. It could be something else. The question is RNA the only outcome, or, can there be others? So now I give you the answer to the CAMEL problem. And so whenever you're not able to solve it, you go to a wise person and then you ask this question, whether this would, how do you solve it? The man says, don't worry, I will bring one more camel. And now he makes it 18 and he reads the will. And now half of 18, he gives the first son 9, 1/3 is 6, 1/9 is 2. And now you add the 9, 6, and 2, that becomes 17. And he takes his camel back. So the question is, are there molecules that come and go that make some transitions possible, which would not be possible if you started only with the pure material? And I'll give you a concrete example now with peptides. So this is what you see, right? Remember the Urey-Miller experiment and Meteorites, which gives you amino acids, that gives you oligopeptides. And then, the question is, people have tried. And if you start with pure amino acids, which is prebiotically implausible anyway, it is very difficult to get to those peptides. So what you do is you think of other things. The reason amino acids don't polymerize to make peptides very well is because they are in this vitrionic form, which you people know very well. It's very difficult to convert this to this and then eliminate water. But what happens if you use alpha hydroxy acids, where you have replaced the NH3 with an OH? Alpha hydroxy acids are very well known. They polymerize very easily by simple drying and heating. And the question is, can we use this, alpha hydroxy acid to help the formation of peptides? And why do we choose alpha hydroxy acids? Because alpha hydroxy acids are present in the same reaction mixtures from the Urey-Miller. They are also present in the meteorites, but we don't give attention to them because we are so focused on amino acids, because amino acid is used in life and alpha hydroxy acids are not used as polymers. More importantly, even if you say I found alpha hydroxy acids, you will never get published in Nature or Science because that doesn't make news. Amino acids is used by life. So what we are doing is we are just using only life as the criteria and looking for chemistries there, which may be actually misleading us. And I'll show you how. So one of the things that, and this is the only chemistry slide that I'll show you, I promise. So the thing is, what happens if you actually start with a mixture of alpha hydroxy acids and alpha amino acids, which is what the meteorites give you, by the way, which is what the prebiotic simulation experiments give you. So organic chemistry teaches you that you will make this ester bond, with the O bond here. And then what happens is, because you have an NH2 group from the amino acid that will make a peptide bond. And you see, you still have a carboxylic acid. And what you do is you will do this repeatedly again and again and again. And before you know it, the prediction based on organic chemistry is you will get an oligomer which is enriched in peptide bonds. Okay, and that is what exactly happens. So this is what we did. So we took alpha hydroxy acids and alpha amino acids, mixed them together, just dry them down, add water, dry them down, just like the earth turning, and then the sun shining on them, drying, rain falling, making the puddle wet again. And then we followed it by mass spectroscopy. And so if you take the amino acid alone, nothing happens. There are no polymers that are being formed. But if you take lactic acid, that is the acid shown here, you get beautiful polyesters forming by the molecular weight that you see. And if you mix them both, you begin to see depsipeptides, where there is peptides and ester acid bonds in the same oligomer. So, and you can do this, and you can do a cycle, and you can follow very nicely. This is the first cycle where you see a lot of lactic acid esters, and that slowly vanishes, and you begin to see peaks that are corresponding to more and more peptide incorporation come in. And if you do this by MS sequencing, where you can go and break the oligomer and then see what the sequence is. The sequence is exactly as what the mechanism predicts. You have nothing but peptide bonds in the center and the ester bonds at the end. Because the peptide bonds are the thermodynamically more stable ones, and they survive this cycling. Okay, so that you begin to see. So I can start with a mixture of esters, acids and amino acids and hydroxy acids, and do this wet dry cycling. And if I do this enough, I may begin to enrich in peptides and may end up with molecules that contain a lot more peptide bonds and begin to behave like proteins and peptides. So now the important question is that alpha hydroxy acid, which comes in and goes away, is your 18 CAMEL. Okay, and there's a beautiful connection to biology that I'll show you in the next slide. That which was very difficult, under prebiotic conditions because of the zwitterionic form, becomes much easier if you actually use the mixture that is in prebiotic chemistry, because it goes through this depsipeptide, which gives you more peptide bonds. And the kicker is, if you know your ribosome, that's exactly what the ribosome does. So that is exactly here, so the ribosome with the tRNA makes an ester bond at the 2' or 3' center, hydroxy, and then brings them too close together, where that beautiful amino group attacks the ester bond and makes that nice peptide bond. So biology does the exact same chemistry, whether it is a historical fossil or biology invented it new. We can't answer that. Remember, I can't answer the historical question. So, and that is also true for the non-ribosomal peptide synthesis, where you use a thioester and do the same chemistry. So that which was very difficult if you focused only on biology becomes actually much easier if you start from prebiotic chemistry. So you can use biology as a clue, but not as the only clue. You have to come from a chemistry viewpoint as well. That's what I'm trying to say. So instead of trying to run the same train of amino acid throughout to the proteins, what you see is now you can do, you start with different type of chemistries with molecules being this and mechanism being the same, but you end up with what biology is using automatically, even without you wanting it. So this is a really non-teleological approach of getting the molecules biology uses, and it's much more intellectually satisfying to see that you have not driven the chemistry, but the chemistry has driven the chemistry. Okay. So what about nucleic acids? Can this work for nucleic acids? So the RNA-world theory, which is the prevalent one, which many people prefer, which in my opinion is not the correct one, is the RNA gives rise to DNA. Somehow proteins, it's like this fairy tale, happily ever after kind of deal. So you go from a homogeneous backbone to another homogeneous backbone. And the question for me was, how does that happen? The RNA world is a construct of running biology backwards. It's not a construct of running prebiotic chemistry forwards. Okay. How does the RNA world give rise to DNA? So people may say, oh, RNA invents proteins. But even if it does invent proteins, the question is, how do you prevent the mixture? There are organisms like yeast organisms which have mixed RNA DNA genomes, gives rise to genomic instability. And you need exquisite proteins to come and remove the RNA from the DNA, remove the DNA from the RNA and keep them separate. How do you do that in a prebiotic world where there were no enzymes or supposed to be no enzymes? And so we did the following. We said, okay, let's take a pure RNA and start doping in DNA and see what happens to these, what we call chimeric sequences, where there is DNA and RNA mixed in the same backbone. And so what we did was we took a pure RNA and started adding in the DNA residues, and we measured the duplex strength stability by UV spectroscopy. And what we found is that the melting temperatures of a pure RNA and a pure DNA are very good, but when you start mixing them, they begin to make very weak duplexes that they fall apart. So that means if you are going to go from a pure RNA to pure DNA, you have a thermodynamic barrier that you need to overcome. You are going through lot of sequences which are unstable in order to get to the other side. So how do you get from a pure RNA to a pure DNA, avoiding this mixture of sequences? So rather than that, what we decided was, rather than going to a pure RNA to a pure DNA somehow through this energetically high barrier chimeric sequences, why not start from the mixtures from the top? Let them make the mixtures and let them go to the thermodynamically more stable structures, right? So we are asking thermodynamics to help us in going from a mixture to thermodynamically more stable duplexes, which is only possible if you have a pure RNA backbone or a pure DNA backbone. The mixed ones, remember, are not that thermodynamically stable. That was the idea. So the idea was, yes, now probably we can start with a mixture, and the kicker is that you don't end up with one RNA system, but you also end up with the other system that was mixed in. So you get two systems simultaneously together, because you need at least two components for a mixture. So that means RNA and DNA could have simultaneously arose rather than RNA giving rise to DNA. So that is what we tested. And so what we did was we did this hypothesis where we said, can we start with RNA DNA mixed sequences? And then that is the proof of principle that we did. Can we start with these mixed sequences? Does that clean up to RNA and DNA? And so what we did was we used an RNA template, and this is a chimeric DNA template. The underscored ones are the DNA. The not underscored ones are the RNA residues. And so we used pure RNA ligands, chimeric RNA ligands, and the chimeric template. And then we used an artificial activating agent, which is used in this field. And we used an amino group because it's much faster in ligating. Otherwise, it takes a long time. So in order for the postdoc not to be for 30 years in my lab, we used this trick to make it faster and see what happens. And the question that we asked was, do you get pure RNA or do you get this chimeric sequence? Which one is preferred when you give this chimeric template? And so what we did is we created a library of sequences and then asked the question based on the chemistry, what happens? And here is the first result. So when we take chimeric templates, which are the templates here, the chimeric ones, the CT, the C standing for chimeric, you see when I give the homogeneous ligands, they react very well and make the almost, and you begin to see that if I use chimeric templates and chimeric ligands, they are much less efficient in making the product. So that means the chimeric template actually prefers the homogeneous ligands than the chimeric ligands. And that is true for not only the AU80 sequences, but also the shorter GT sequences. There is a temperature effect that you see. And so we ask this important questions. In RNA world, the theory is RNA replicates itself. But there is a big problem. What is known as a template product inhibition. That is, once the RNA ligates and makes the product and the template are so strong that they don't come off. Only if they separate, you can go for further replication. But since this is too strong, it's known as a template product inhibition, and you just produce 100% and you stop. You don't multiply further. But with this chimeric template, which is weaker, we thought maybe if the chimeric template could separate, maybe it could go further. And that's what we tried. So we took a chimeric template, gave it pure RNA ligands, and then asked whether it would make this duplex. And since it's a chimeric pure RNA duplex, it should separate if you give the complementary RNA ligands to the newly formed RNA product, and it should take it away, releasing the chimeric template for another cycle of replication. And that is what is shown here. So you see almost 200 to 250% of product formation of the RP3, which is this product right here. And the RP2 is formed in very good amounts as well. So you begin to see how the chimeric template begins to act as a catalyst because it's being released back into the reaction mixture. Now, how does this compare with pure RNA template? And so that's what we did. So when you give the pure RNA template, it can also do this. And we found that every time the chimeric RNA DNA template outperformed the pure RNA template. That means you are better off starting with a mixture of RNA and DNA in order for you to replicate rather than starting with pure RNA or pure DNA. So that is the idea. And then, so you can do this with mixture of DNAs. It works the same way. And then we ask the question, if you put a selective pressure, what happens? Like you begin to dilute, something else comes in, which performs better, the pure RNA template or the chimeric RNA template? And every time you see that it's the chimeric RNA template, DNA template, that outperforms the pure RNA template, as you see from this HPLC trace, where you can see the product forming when you use the chimeric template over the RNA template. So you can get into an RNA world, but you can't get out of it, because the RNA world is kind of a dead end for you. But if you start with the mixture, then you can get pure RNA and pure DNA, the same thing that you would want. But you can start with a mixture instead of a prebiotically implausible pure RNA, chemistry. So you can also lead to cross-catalysis. So the question that we asked was, okay, let's start with the chimeric template and make this, but instead of giving pure RNA ligands, what happens if you give the chimeric ligands? Do you produce more of your chimeric template so that it can become amplifying? And that's exactly what it does. So you can see this is the original one that we found when we don't add anything. But when you add the chimeric template, you see now compared to the original, you begin to produce more of your product, because you are producing more of this chimeric template in the reaction mixture. So that means starting with the mixtures actually gives you the solution that you always wanted, but you couldn't achieve when you started with pure compounds. So that's exactly what we are trying to do. So this paradigm that I'm showing you here, that starting from mixtures can actually give you, and the question that you find is, because these chimeric strands go away, they are acting as the 18th camel. They come in, they solve the problem for you, and the wise man, if there is a wise man, takes it away, and you end up with pure RNA and DNA. So that was very intellectually satisfying to see that you can actually start with mixtures and the solution pops out naturally without you directing the solution. Okay, and so the implication is that you get two systems at the same time, not one system, which are already in communication with each other, so they don't have to invent and learn about each other new. And the implication is I can use messy prebiotic chemistry, a systems chemistry approach, rather than focusing only on the synthesis of pure RNA or pure DNA building blocks. And now there are many research groups that are trying to make the RNA DNA building blocks together. You might have seen a famous paper from John Sutherland in Nature recently, where they show how you can also make the DNA building blocks the same type of chemistry that they use for RNA building blocks. So the field is slowly coming to the idea that the pure RNA world might not be the best way to understand how life arose on early Earth. So the same way, you don't have to drive RNA all the way throughout. You can start with chimeric backbones and move over to homogeneous backbones as the chemistry progresses. Okay, so what this opens up, interestingly, and I will finish in about 5 minutes or less than 5 minutes, is that this allows us to see if the early Earth was different. Remember, our biology is contingent upon water, 0 to 100 degrees temperature, and a pH of around 7. What happens if the pH was always around 3? We would not have had the same system that we had today, because Watson-Crick base pair will not function at pH 3 or pH 12. You would have to come up with different chemistry. So if the conditions were different, you could have had alternative life on Earth. What would that alternative life look like? And I will try to give you an idea based on biology. You may have a different biological paradigm, and I'll give you one example. Okay, so what we do in our group is, we work with RNA, but we also work with molecules that simulate or look like RNA. So this is one made on ribulose, not ribose, ribulose and xylose, which is found in biology. Incidentally, nature does not make ribose straight away. It makes ribulose, xylulose, and converts them to ribose. Okay, that's something to keep in mind. So we looked at this ribulose and ribulose, and you see the bases are on the same side as the phosphodiester backbone. Here is the basis on the opposite side of the phosphodiester backbone of RNA. So keep that in mind. And when we made these oligomers, we found that there was no base pairing, because when you try to do a double helix with this, the phosphates are on the same side, so they are much closer, and there is a electrostatic repulsion that pulls them apart, and therefore we didn't find any base pairing. And when you compare it with now RNA, you see beautifully how nature has maximized it. This is the farthest you can get away with the phosphodiester backbone, and the closest you can be with the bases. The bases are inside hydrophobic, the phosphates are outside hydrophilic, and it's a marvelous arrangement in water where you would get this duplex structure. Remember, you don't get this duplex structure in organic solvents, only in water. So water plays an important role. So now the question was, so these things don't base pair, these things don't base pair. And we wanted to understand, the synthesis is difficult. So I asked one of the postdocs to make this molecule. This molecule is, there it is ribose, and here is xylulose, ribose, xylulose. So it's a alternative repeat units of ribose, xylulose, oligonucleotide. And he made it, and then we wondered whether they would base pair. But remember that, as I told you, xylulose has no base pairing. It doesn't base pair with RNA. And so here we have 50% xylulose in this backbone. And the question was whether it base pairs or not. The prediction would be, no, how can it base pair when xylulose has no base pairing with RNA? But surprisingly, we found that it formed beautiful structures of a nice duplex with base pairing. And how does it compare to pure RNA? It's almost as strong as pure RNA duplex. So that was a big surprise. That means you are mixing two systems which have no base pairing, and then you are creating a system that is almost as good as RNA. And that taught us something. That means we are spoiled by biology, and we are not looking for anything else except biology, because that's the only thing that we know. So we proceeded further and said, okay, so if we can make these mixed backbones in an alternating fashion and create new systems, what other systems can we create? So here is a ribose system. So the blue one is this ribulose structure, which is a pentulose. This is a pentose. So this is an aldose, that's a ketose. And you see, you can mix them in an alternating fashion and create systems that are as good as RNA. That means a heterogeneous backbone with different bases can give rise to informational systems. And I will end up with this slide where I show you how you can change the Watson-Crick paradigm. You have been taught in your life all the time that GC is stronger than AT because of the 3 hydrogen bonds. That's not correct. It's because of the hydrophobic nature stacking, not because of the number of hydrogen bonds. And I will show you why. So what we did was we took this mixture where we have now 2 different sugars in the oligonucleotide backbone. So the ribose here has the pyrimidine thymine, and the ribulose has the purine adenine. And when we interchange them, and now here is the same thing, guanine and cytosine. And you see GC is stronger than AT. That's exactly what you would expect based on biology. But if we switch and we change now, put the cytosine on the ribulose and put the adenine on the ribose and do the same thing here, now you see your AT is much stronger than your GC. So we have completely flipped the Watson-Crick paradigm and created a system where the 2 hydrogen bonding AT is much stronger than a 3 hydrogen bonding GC. So I can now come up with an oligose nucleotide which contains only 2 nucleobases, thymine and adenine, but with 2 different sugars. And depending on which sugar carries which pyrimidine or purine, with the same AT, I can simulate a GC or an AU. Okay, so that means I can change the Watson-Crick paradigm by changing the backbone. And that teaches you that the AT-GC paradigm that you have from Watson-Crick is only possible because you have a homogeneous ribophosphate backbone. We forget the backbone, we focus on the AT-GC, but that is entirely dependent on the backbone of the RNA that you're using. Okay, you change the backbone, you change the rules. So I'll end up in a couple of minutes. Do not focus in order to understand how chemical evolution gives rise to life, do not focus only on biology and therefore only on that component in prebiotic chemistry. Do not extrapolate biology all the way back to prebiotic chemistry, and the other way. Do not extrapolate prebiotic chemistry all the way to biology. Okay, and look at what other molecules would have been present. That is the reality of prebiotic chemistry. When you say, I found amino acids in meteorite, there are other molecules also on the meteorites, but you focus only on amino acids because that's what biology uses. But that might be a mistake in understanding how biology came about when you ignore other molecules. So other molecules could have helped, as I showed you in 2 examples here. So the question is, if you believe in this, you can also believe in chemical evolution, And so I end up with, thanking my research group, the funding agencies, my collaborators who worked with me in the depsipeptide project. And I leave you with this philosophical slide to keep in mind whenever you read a research paper in this field, whenever you see life found on Venus, please, do not rush to that conclusion, because we can fool ourselves very easily. Case in point, when this Martian meteorite, Alan Hills landed, people thought, worm-like structures, and they said, oh, life on Mars. So let us be careful and be grounded in chemical reality to understand how our biology came about. So with that, I finish my lecture, and I'm happy to answer questions.
Thank you, Ram, a lot for this very beautiful and passionate talk. I'd like to remind or point out to our participants that in handouts, he attached some articles from his research, as well as a Quanta magazine, in case you're curious about. Can you hear me?
The volume is very low for I do not know why the volume from your side is very low, because I've kept it fully on my side, and it seems to be quite low. I hope people were able to hear me well.
Is it better now? Still not?
Yeah, that's okay. I can still hear you, no problem.
So if then I will just move over to the questions. So the first question is from Panos. So he's asking, RNA is a very interesting molecule. Do you think that modified RNA bases are remnants of intermediates that happen to be functional in the polymer, like orotic acid, or later additions in evolution?
Yeah, very interesting question. The ones that are now in tRNA that we see seem to be post-translationally modified. That means biology seems to have created it. But there are people who are, working on the assumption that there are bases like cyanuric acid and triaminopyrimidines, which would have given rise to RNA-like structures, but then are not present in biology anymore. So the modified bases that you find in biology today seem to be a post-translational modification. However, there is a group in Munich, led by Thomas Carroll, who's working on the possibility that these modified bases might actually have been present in ancient RNA and then might have been resurrected in this post-translational modification because they are functionally useful. So this is an answer that can be answered, this is a question that can be answered by actually doing the research and finding out whether they do give rise to functional systems that would lead to RNA, So, but that doesn't mean that they did. As I said, this only shows it can happen. So with that caveat, I would say, yes, that's something that we should consider.
Thank you. So the next question is from Rashi, and she's asking, have you ever considered and tested the possibility of PNA, peptide nucleic acids, as a starting material for all DNA, RNA, and proteins?
Yes, we have. And we used the true peptide nucleic acid. The one developed by Peter Nielsen is not a true peptide nucleic acid. If you go and look at the structure, it doesn't have peptide bonds repeating. It has peptide bonds and an amide bond in between. So it's not a true peptide bond, because it has a diethylene unit in there. And we studied peptide nucleic acids as well, and we found that it has its limitations. And the type of nucleobases that you can attach to it, by the type of chemistry, you can give rise to, you can construct peptide nucleic acids which are base pairing. But then the question is, once you make it and you show it's functional, can you also generate it in a prebiotically plausible way from the molecules that would have existed on early Earth? So to answer your questions, not only PNA, there are other systems that would do the same thing that I discussed. But how do you generate them in a prebiotically plausible fashion is the sticking point. So I can generate PNA, and I can show it base pairs, and I can show it acts as a template for replication, etc., etc. But the PNA that we have and we have tested, the problem is because the backbone is uncharged, the product inhibition that I was talking about in replication is greater for a PNA than for the RNA. So it also runs into the same problem of an RNA world theory where it cannot replicate itself. So that is why the PNA research has not proceeded well until somebody comes up with a solution to that. PNA was considered very strongly, and it was favored one time, and people invested heavily in it. But the problem of generating in a plausibly prebiotic fashion and then replicating it became a problem within the context of origins of life studies that people abandoned it.
Thank you. So I think we have time for one more question, and Rohan is asking, at what time point do you think chimeric sequences disappear from the cell, resulting in pure nucleic acid sequences? Would it possibly have to do something with coevolution of the requisite protein machinery, which could potentially fill in for the function for these?
Yeah, I would agree with the second part of your answer. That means there must have been another machinery in place that could compensate and be much better than what the chimeric ones are doing. So if you look at what I showed with the chimeric sequences, it has its limits because it can't produce very long sequences. We are working with only short sequences. But what happens if you go to longer sequences where the product template inhibition would be even more severe? So you are absolutely right. Coevolution, and I pointed out in our paper where I have deliberately omitted proteins and lipids in my experiments, but maybe including them will give us more answers and more insights into when that transition that you talk from chimeric control to protein control can take place. Agreed.
Okay. Well, thanks a lot for your talk and for taking your time to answer our questions. I would just quickly like to remind the participants who have assisted any of the posters to vote for your favorite poster, as well as a reminder that at 8 p.m. CEST, we have our social event happening. And again, thank you, Ram, for joining us today.
Thank you for the opportunity. Enjoyed it very much. Thank you.
Thank you.